All right, folks, so we're taking a look at Part B of Chapter 9 and moving on from Part A. And really what Part B is discussing is taking the information that we have discussed in Part A, muscle contraction, the theory of how muscle contraction works through these individual functional units called sarcomeres, and then taking that information and applying that in a larger scale. And so we're really looking at how muscle groups operate and how they actually undergo contraction. Again, we have to keep in mind that this is because of those individual muscle. And so the same principles that we use to study contraction in a muscle fiber, we also look at when we apply that to a whole muscle. And again, this is our focus in Part B, is looking at whole muscle group functioning as well, right? Contraction produces muscle tension, right? And what tension is the force exerted to move a load or an object, right? Force applied to move a load or an object. And what would that object be? Many times it is actually, bone, tissue, etc. But sometimes we have, and all of that applies to, like, let's say a limb, right? So if we're going to use muscle to move a limb, but we can also then, add on weight to that limb. Like, for example, if you were to lift something using your hand, if you will, whether it be a cup of water or, you're moving a big couch or something like that, that weight of that object plus the limb, which is all the bone and tissue, is actually moving, using the muscle to do that, right? So that's really what they're talking about here. Now, when we talk about contraction, we took a look at how muscle shortens, or how the contractions shorten. But in fact, when we're looking at a whole muscle, muscle may or may not shorten, contraction may or may not shorten the muscle. And what we call isometric contraction is actually not shortening the muscle. The tension increases, right? So there is contraction, okay? But it doesn't exceed the load. And because of which, we see much of a stretch. Much of a stretch. And this is, in fact, if you relay that back to, going out to a gym and working out with weights or what have you, there are types of exercises in which you can actually use a weight to lift and not lift, right? Lift and stop, like you are using a dumbbell. And that causes movement. And in fact, the muscle does shorten there. You would call that isotonic contraction, okay? Again, using a dumbbell, right, will shorten a muscle. But there are some exercises, in fact, if you go to a gym, where you actually, all you do is lift the weight, or, hold the weight in your hands or hold the weight in your legs, and then keep that contraction steady. You don't need to actually shorten the muscle or anything. You keep it right, steady, like a deadlift, right? So if you know what a deadlift is, when you lift weights, that's a great example of that as well for an isometric contraction. So it doesn't do any shortening. It actually just keeps the length of the sarcomeres in place. Tension is there, but it doesn't cause shortening of the muscle. Okay, the force and duration of the contraction is going to vary depending on how much stimuli, okay, or the intensity of the stimuli, right? So again, what are we talking about? What is really this, are we discussing here? The nervous system. And again, as I mentioned in Part A, the nervous system is directly related to the muscular system, right? The nervous system stimulates, tells the muscle, okay, time to contract or time not to contract. And again, we can actually change the stimulation, how much stimulation we have, right? By how much we stimulate the muscle and how strong we stimulate the muscle, right? So this means how much stimulation we're doing and how strong. Okay, that's what frequency means and intensity means, right? So again, the nerve can operate this way, and because of that, we can actually increase the amount of contraction and how long we contract in a muscle. That's really what we're talking about here. Each muscle is served by at least one motor neuron or one motor nerve, and in fact, there's many muscles that actually have different nerve innervations. There's different nerves innervating it, right? One motor nerve can contain axons of hundreds of motor neurons, right? So one nerve is actually, for the most part, a bundle of axons, which is part of a motor neuron. Okay, they're bundled together, and this is what we talk about when we talk about a nerve, right? Now, each of those axons will branch into many terminals, and each of those terminals forms that NMJ, right? The neuromuscular junction, right? With a single muscle fiber. And that's what we're seeing there, yeah? What we call a motor unit, again, as a, to revisit this, is where the nerve and the muscle function together. And again, if I drew that area there, this would be the synaptic end bulb, right? And this would be the muscle fiber. And lo and behold, this area right here would be the neuromuscular junction. That's what they're referring to there. Okay? Again, you have your synaptic vesicles here with your acetylcholine, and again, through all those step processes, then we can create an action potential in the nerve sarcolemma, causing then stimulating further contraction, right? Okay, so motor unit is a motor neuron and all the muscle fibers it supplies, right? So we can go as low as 4 to several hundred, and in fact, one neuron can branch out to many different muscle fibers, right? So we can see this happening, right? Now, depending on what motor neuron it is, okay, the smaller the fiber number, right? So again, we're looking at smaller the fiber number, the greater the fine control. Okay? So what do we mean by fine control? This is fine muscle control, right? So we know this to be true. Again, look in your own body. Some of the movements that you actually use are fine muscle control, right? For example, what I'm doing here in the fact of writing, right? Eye movement, right? And things like that, right? Using those muscles in your mouth to actually push air through to tighten the connective tissue so that you can speak, right? So all those are examples of fine muscle control, right? Other control is, gross muscle control, big muscle control, but we're talking about the less number of fibers that neuron innervates or stimulates, the finer the control. Okay, so this is what we're looking at. Here's our example of what we see as actually 2 motor units. Okay, and so before that we see this is our spinal cord here. This round circle is our spinal cord. Out of our spinal cord, we have what we call a nerve. And in fact, this area here, this area here is our nerve, right? That's our nerve there. And in fact, a nerve is a bundle of neurons. And in fact, there's actually two here, right? Motor unit 1, motor unit 2. And these are motor, so we're having the outputs going out, right? The signals coming from the brain to the spinal cord, down the motor neurons to the muscle to innervate them. And you'll notice that at the synapses, right, there's actually many branches, right? So we've got many branches here serving individual muscle fibers. So in fact, you know, this one serves this muscle fiber. 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 And in fact, I should draw the arrow both ways, right? This one serves this muscle fiber. And lo and behold, this is what we're seeing, right? And in fact, when you look at a photomicrograph, these black dots right here represent the neuromuscular junction. Okay? All right, so what is a muscle twitch? All right, a muscle twitch is a simplest contraction, right? This is one muscle fiber's response to a single action potential from the motor neuron, just one response. And actually, a muscle twitch is what we've been looking at in Part A, one muscle twitch. One muscle twitch results in the muscle fiber contracting. Many of those sarcomeres contract, and then they relax. And so when you actually look at a piece of equipment that we call a myogram, this piece of equipment is attached to actually the body, and will actually read this contraction occurring, right? This muscle twitch. As a result, what you'll see on the screen is that you'll see movement of the graph representing when the muscle fiber contracts and when it relaxes, forming this twitch. And that's what they call tracing, right? When the graph starts moving up and down in just a second. And so there's three phases of muscle twitch, right? The latent period, the period of contraction, and the period of relaxation. Okay? In the latent period, based on the graph, this is where you actually see the EC coupling cycle. This is the stage. This stage sets up the events for actually contraction. And if you recall, then the EC cycle, this is where the acetylcholine gets released. The receptors bind to the acetylcholine binds to the receptors on the muscle. We have those ions getting released from the ion channels. And then we start to see an action potential generated, travels down the sarcolemma or sarcolemma, right? And then we see that innervating the SR to release calcium. And so all of this is part of the EC cycle, because of which this stage we don't see muscle tension, because we don't see contraction quite yet. We're setting up all of those stages that we have discussed in Part A, all fall under the latent period, right? Where we don't see on the graph any muscle tension, right? And again, what that really means is that the EC cycle is occurring, right? And in the period of contraction, this is actually where we see cross-bridging, where those myosin heads bind to the actin molecules. And again, this is where we see troponin, tropomyosin. Don't forget that. That's part of cross-bridging, where tropomyosin, troponin help to open the active site of actin. ATP gets used for the myosin head to attach themselves, and that's where cross-bridging forms, right? Cross-bridging forms. Because of that, we start to see the myosin heads move and tension increases. So again, latent period, no tension. Period of contraction, tension. And then the last phase is where we see relaxation, where calcium actually gets re-entered in the sarcoplasmic reticulum. And in fact, it gets pumped back. And we pump that back using a molecule of ATP. And so what this really means, because it doesn't tell you that the calcium gets pumped back, what also happens is the heads detach from actin, troponin, tropomyosin, slide back in place, heads cock back, and it is and the muscle fiber is ready, or the sacromeres are ready for another contraction to occur. Okay. So we see this occurring if we take a look at a myogram, which is, again, if I attach this to a muscle and then watch this happening, we see then time at the bottom. This is your x-axis, and this is your y-axis on a graph. And then we see tension on the y-axis, and we see stimulation at time 0. So when we start the clock is when we hit the stimulation of the muscle. So we do this on purpose to the muscle and then watch contraction go up. And so you can see contraction go up and then fall down. Right? So it's only one stimuli, by the way. It's only one stimuli. So we haven't re-stimulated the muscle. We just put a synthesized action potential and then watch to see what happened. And then, of course, this really small area here is the latent period where you don't see contraction, and then quickly rises to here. And in fact, this is where you see the period of contraction and the events of the period of contraction. And then finally you see this here, which is relaxation. Right? And the graph indicates that as well. You'll notice that it contracts much faster than it does relax. So that means that it's a much shorter time to contract. So it's much faster than it is to relax. So you see this here, right? Much longer time to relax. Right? And that's normally what you see. Again, that falls in line for the function that it contracts really fast and relaxes, but that relaxation takes much longer. Okay, so how strong a muscle twitch is and how long it lasts is actually due to different metabolic properties and enzymes between muscles. So some muscles have different metabolistic properties. They undergo metabolism differently. They have different enzymes, and because of that, their strength varies and their duration of the twitch, how long the muscle contraction contracts, right? So great examples, right? The eye muscle contracts rapidly and brief, right? So this is the muscle that moves your eye left, right, up, down, and all angles through, right? And heavier muscles like the calf muscle contracts slowly and holds it for longer. So again, the strength and duration of these two muscles vary completely. So we're looking at two different extremes here in the. And in fact, if we took a myograph, right? Okay, like we're seeing here, and we took a myograph of the one of the muscles of the eye. This is the eye, one of the eye muscles, and then we took a look at two muscles that participate in the calf muscle, in the calf contracting. So these are calf muscles, and we took a look at each one of those, and we stimulated them separate from each other. And then we took that information, those graphs, and we put them one on top of another. We see that the eye muscle contracts really quickly and then relaxes really quickly, right? You'll see the gastrocnemius muscle contract and takes longer to relax. And then you definitely see the soleus muscle do this, right? Takes much longer to contract and holds it for much longer and then relaxes much longer, right? Okay, so let's look at a graded response, right? Muscle response, okay? So normal muscle contraction is usually smooth. The strength varies depending on what we're going to use it for, right? So there's a big difference between lifting, let's say with your hand, a pound of something versus, I don't know, 20 pounds of something. So it's going to vary depending on, you know, how much of that muscle we need to use for contraction to lift that object, okay? So when we see a graded muscle response, right? What we mean by that is we change the strength of contraction for different demands. This is what we're talking about. This is what a graded muscle response is. Graded means in different stages, right? So if we need to use just a little bit of muscle, that would be one stage. But if we need to actually use more and more, we can recruit more, or we can stimulate more muscle fibers. Or if you could use the word twitch, we'd use more muscle twitches to then contract more muscle fibers to cause then an even stronger force to use because we're using more weight, right? So we do two things, right? We change the frequency and the strength, right? This is how strong the stimulation is, right? That's what that means. And how many times do we stimulate? That's what that really means. Frequency and strength, that's what they really mean. Okay? So we know this, right? Single stimulus, single response. That's your muscle twitch. That's your nice in a lab if you wanted to use a myogram and test this out, you know, one stimulus, one muscle twitch, right? Normally, in reality, it doesn't work that way, right? Our muscles are always twitching, right? So we get a lot of twitches at the same time. But again, that's our individual. So what we see, for example, when we see two stimuli, one after the other, so if we increase the frequency, right, here. So if we increase the frequency here, this is what we see. We call this a wave or temporal summation, right? Let's say it's two stimuli in rapid succession, one after the other. And because of that, the muscle fibers don't have time to completely relax, right? So the twitches, in fact, in response, the twitches increase in force with each stimuli. So what is occurring in the sarcomere, in fact, and what's occurring in the SR is that more stimulus that stimulates then those channels in the SR cause additional calcium to be released. And that happens with the second stimuli or even a third stimuli, and so on and so forth. So if you decrease the time of the stimulus, increase the frequency, that secretes more calcium. More calcium causes more contraction to occur. So look at this, right? Here's our first stimuli in this arrow here. We see then, here's tension there for our first stimulus, right there. Okay? So let's say we decrease the time. We say, well, I'm going to hit the second stimulus. Okay? It doesn't have time for the muscle to completely relax, and this is what we see. Right? So tension is increasing. You can see tension going higher and higher. So then we hit it a third time. We see this occur here. And then let's hit it a fourth time, and we'll see it here occurring. Again, if we hit it a fifth time, we would see probably another rise here, and then there's a certain limit, right? So we'll hit a limit. We'll hit a ceiling, if you will, for contraction. So even though we apply more and more stimuli closer and closer together, we'll get to a point where, you know, there'll be just this happening, right? There's only so much a muscle can contract, and we have a limit to that. And that's really the strength of the muscle. So again, if the frequency increases, then tension's going to reach a maximum. And we call this contraction, this red line that I drew here in the previous graph, summation, right? And then further increasing is really getting to that point where we have a sustained quivering contraction. We call this an unfused tetanus. Okay? Yes, we use the same word as, for example, tetanus shot or having tetanus. This is actually where that disease name came from. It's not the other way around. It's actually the disease itself, tetanus, that comes from an infection, a bacterial infection. Actually is because we use this name tetanus to describe this type of phenomenon, this type of action here. So here's what you see, right? So keep in mind, this is what you're really looking at is the stimulus in a really short period of time, right? And as we do that, we start to see an increase in tension, and at a certain point, we start to see this unfused tetanus, where there's this wave and wave and wave, and this is where you see this kind of contraction almost continuously. Now, if you increase the frequency to a certain point, then it reaches a maximum. That's what I'm discussing, what I was referring to in the red line, and then we call this a fused or complete tetanus. So it actually looks like one complete line, right? Okay? And keeping this contraction at this stage for a long period of time can lead to what we call muscle fatigue, okay? Or muscle weakness, right? That's not a permanent thing. This is actually a natural process, right? Where the muscle stops contracting, and then we'll have a long period of kind of recouping everything because it's been used very frequently for our sustained time. And this is what you see, right? Notice the stimuli are very, 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 very close together. Because they're very, very close together, we start to see this kind of rise, and then this maximum here. This is the maximum line for this muscle here, right? That's your maximum. All right? And then we see this sustained contraction here. Again, we call fused tetanus, right? Or complete tetanus. And again, that's the maximum amount of contraction that muscle can actually undergo. Okay? So we use the word recruitment, okay? And recruitment is sending different stimuli to different muscle fibers. So again, we took a look at, let's say, just one muscle fiber doing this complete tetanus. But before we get to that point in reality, before the muscle fiber that one muscle fiber gets to that point, we're going to try to recruit other muscle fibers in the area, in that muscle group, to then start to have more stimuli and more contraction there, right? That leads to more precise control. This is what happens when we actually have this fine control, for example, when we use these muscles. Okay, this figure just really shows then the stimuli, right? How much stimuli, 1 versus 10, and then how much strength of the muscle there is, right? So the more stimuli you have, the more then strength you're going to have, right? And I just want you to look at this. This is kind of really important to look at, right? If you look at this, you can see one muscle fiber. So this is, all these circles represent the muscle fiber, and it's just one over time. And what we're seeing here is one stimuli to one myofibril. Okay. And then another, and we can see where it starts to turn from this no color to then colorized circles. That means those are getting stimulated, right? And then over time, you see the last 4 there, all being stimulated, all the muscle fibers being stimulated. Okay, so recruitment works on, because that's what we're talking about, on the size principle, whereas the motor units with the smallest muscle fibers are recruited first. If we need more, then we'll use the larger ones, are recruited second and third. And the largest motor units are only activated for the most powerful ones. So by size. So this works asynchronously. So that means that some fibers can contract while others rest. That helps prevent fatigue, muscle fatigue. And this is what we see, right? This is one small motor fiber being used first. And then if we need more contraction, more strength, we'd use the medium ones. And then if we need even more, we'd use the large. Okay? Look at the skeletal muscle fibers, which tells you that some are this size, some are that size, and some are even bigger. So their sizes can vary. Their muscle fiber sizes can vary. So you might want to ask yourself, can I change these into these and these into these? And the answer is yes, you can. So you can make larger and larger muscle fibers, but you cannot create muscle fibers. You can't create them. They're actually already there. So as far as getting bigger, what we call getting bigger, but increasing muscle, what we're increasing is actually the size of the muscle and not the muscle number. Okay? So number of muscles doesn't work this way. We can't actually increase the amount of muscle numbers, but we can increase their sizes. And the bigger the sizes mean bigger muscle, and that's what you do when you actually look at, making bigger muscles, looking at bigger muscles from the outside of the body. Okay, so what is muscle tone? Muscle tone is actually a consistent, slight contraction of all muscles. Okay? And this, in fact, is because of the motor units. They're alternately activated in response to input from stretch receptors in the muscles. Okay? So what this means is your muscle itself is going to be, your muscle yourself is going to be innervated. When we call innervation, that means it's going to be read by these receptors on your muscle fibers, and actually not on just the fiber, but the muscle group and the ligaments and the tendons. We have these receptors we call stretch receptors, and they actually stimulate in response to muscle getting moved, contracting, movement occurring. And what they do is actually report that information back out, okay? And we call this the input, right? So what this line represents is actually a nerve responsible for sending information, let's say from muscle to then the spinal cord and the brain, ultimately the brain. So the nerves connected to the spinal cord, and then that information is relayed back up the spinal cord to the brain itself and neurons in the brain. And so the brain will make a decision then, okay? And when we talk about a decision, they'll send the signal back down and then send that information out to those motor units, and we would call that the output. Okay? So output's also known as the motor response, and input is known as the sensory response. So we're sending information to the spinal cord and brain. Brain makes a decision, sends that information out. And when we're talking about a muscle tone, these motor units are alternately activated. So some will be activated consistently and some will stay dormant to then always have this nice contraction of the muscle. Because we have these contractions of these muscles, we keep the nice little firm, healthy type of muscle that we know to be muscle tone. When we talk about, being healthy, working out, exercising to then have some nice muscle tone, it's actually because of the muscles, individual muscle fibers are always contracted, keeping the muscle nice and firm, right? And that's what you see there, right? Always relaying information and then sending the information that causes muscle to stay contracted. Okay, well, so what do we mean by isotonic contraction? This is the one that actually shortens the muscle, changes in length, moves a load. That's what we mean by isotonic contraction. A concentric contraction is actually the shortening of the muscle. Again, picture your bicep, using your biceps. Eccentric contraction is actually the lengthening of the muscle. And that generates a force. Think of your muscle not lifting stuff, but let's say, putting something down. It lengthens. We still have a force generated, and that's still considered contraction. Okay? So we see this, let's say in research, we take a muscle like that, and then we stimulate it to cause a contraction. We see the muscle shortening, obviously. Right? That's mainly concentric isotonic contraction, right? Isometric contractions, the load or what we're trying to lift is greater than the tension of the muscle. So the muscle doesn't short or lengthen, but stays in a contracted state, whereas it stays in that, original length. Okay, so let's talk about energy really quick. We need energy for contraction. We use that energy in the form of ATP. Okay, So what do we use that ATP for? Cross bridge formation, when the heads detach, when we need to pump back calcium, which we do when in the relaxation cycle into the SR. We actually use ATP to pump out sodium and pump back potassium after the excitation contraction coupling. And again, this is from the action potential stimulating the sodium channels to open, the potassium channels to close, and then we've got to repump that back, getting everything back to the relaxation phase, right? Okay. So as I mentioned, we can only store ATP in a very small amount. In certain areas like muscle, we can store ATP, but it's not in a great amount. And in fact, when we first contract muscle, we only have about 4 to 6 seconds worth of muscle in our muscles. 4 to 6 seconds of ATP usage in our muscles. And so because of that, we need to consistently generate ATP, right? So it has to be regenerated really quickly. It has to be made really quickly if we want to continue to contract muscle. Okay? So we do it in three ways, right? ATP in 3 ways as it pertains to muscle. We phosphorylate ADP by this chemical that we call creatine phosphate. Okay, creatine phosphate. And for example, a lot of stores, a lot of health companies actually use this as a nice selling tool because they sell a lot of this in a powdered form, creatine. And they tell you that creatine is really important for muscle building and that we use creatine to actually build muscle. But it's one of those marketing techniques that, although it's true, you know, part of that is true. We do use creatine to actually contract muscle. The rest of it is false, right? So, and in fact, you don't need the amount of creatine even in the amounts close to what they sell you, right? So a lot of that gets taken in. If you decide to use it, a lot of it gets taken in, and then we basically, you know, take that out of our body in the form of our, you know, we poop it out or pee it out, basically. So it's a good waste of money for the most part, right? So I'll talk about that in just a second, how creatine gets used. We anaerobically make ATP through glycolysis and lactic acid formation. I'll talk about that. So these are ways that we make ATP, okay? And the aerobic pathway. This one right here is the most preferential one. This is the one we want to actually use most of all, and actually that we do use most of all. This one right here. Okay, so creatine phosphate is actually a really special molecule. It's found in muscle fiber, and it actually is the one that, since it has a phosphate group, will donate phosphate to ADP and then make ATP from there, right? There's an enzyme called creatine kinase, and that's the enzyme that actually transfers the phosphate to ADP and actually makes ATP. So we actually use this method of making ATP in our muscles, and that method can only give us about 15 seconds worth of ATP if we're contracting and relaxing the muscle. Okay, let's say you start off running. You say you want to jog, 4 miles this afternoon, and you start doing that, right? And the first, couple seconds of using the muscles, your leg muscles, to contract and relax, that ATP comes, that first 15 seconds comes from this pathway, right? So we look at ADP, right? Stands for adenosine diphosphate. And it's actually a reusable molecule. So we don't create this molecule in this process of generating ATP. In fact, all we do is add a phosphate group, and this little line represents a phosphate that's by itself. to create ATP, right? And then we take that ATP, and when we break the bond of that last phosphate group, we release energy, and that ATP becomes ADP, right? And so when we do that, we release the energy with the phosphate again. It becomes ADP. And then we do something like creatine phosphate to add another phosphate group, creating ATP, the molecule holding energy. So I got to mention that ADP doesn't get, that's where that ADP comes from, because it gets reused over and over again. And again, seconds, right? You see ADP here, you see the enzyme here that carries the phosphate group from creatine, creates ATP, right? There's creatine phosphate, CP, and there's creatine, right? Free creatine. Again, 15 seconds. All right, so here we talk about this being the anaerobic pathway. Okay. Anaerobic means no oxygen. So actually this process does not need oxygen for it to complete the reaction, right? So again, how do we make ATP, or how does ATP get generated here? We actually break down the energy storages in the bonds of glucose. So I will draw you this, right? Which this line represents the bond between one carbon group and the other of glucose. Right? This is glucose. And in fact, when we break this bond, or when we break the bonds of between the carbon and carbon group, we actually then release energy. Okay? The first process of breaking all these 6 carbon bonds, releasing energy, we always start with the process of glycolysis, right? Which stands for glycolysis, which means glucose getting broken up. And again, we use the first process here to break that down. And again, it doesn't require oxygen. Glucose gets broken down into 2 pyruvic molecules. These are what we call intermediates, but these are molecules that are a result of breaking down glucose. And what do we generate? Only 2 ATP doing this, right? So it's not an efficient way of breaking down glucose to harness energy. And in fact, 2 ATP molecules is pretty small, right? Okay. If we have low oxygen levels, for example, if we don't get enough oxygen in our tissues, pyruvic acid builds up, and it can't get broken down even further, because we can actually break down pyruvic acid even further and harness more energy, and that doesn't happen in a low oxygen level. It gets built up. And if it builds up, right, normally pyruvic acid goes to the mitochondria of your muscles. Do you remember that mitochondria is there next to your myofibrils, right? And mitochondria actually use pyruvic acid, right, and oxygen to break the further break down pyruvic acid into more energy, right? Normally speaking, pyruvic acid can be broken down that way if oxygen is available. If it's not available, in fact, when you don't have enough oxygen in the muscles, pyruvic acid gets converted into lactic acid. And lactic acid, in fact, leads to soreness and cramping, right? And that's the side effect of actually using muscle over time, right? And that can be relieved. The more the muscle gets used, more oxygen gets taken, more blood vessels get made to take the oxygen there as well. Lactic acid also can be taken in the bloodstream eventually, so it does take a while for this to happen. So that's where the soreness and the the cramping can occur after, a strenuous workout, right? So lactic acid can be used, and it usually gets taken by the bloodstream. Again, takes a while, and then we can use that as fuel in those places. And actually we can convert it back into pyruvic acid by the liver. And we see this here, that glucose gets broken down in the process of glycolysis. You see the pyruvic acid there, you see the lactic acid, but then it can still generate ATP, which is not a lot, right? And this actually gives us energy for the next 30 to 40 seconds of muscle contraction. Okay, so the anaerobic pathway. Again, that's a very short amount. The one that does produce a lot of energy is aerobic respiration. That's the one we prefer, right? 95% of the ATP at rest and light to moderate exercises comes from aerobic respiration, right? Although this is the one that's most preferential, it is actually slower. It takes a slower amount of time than the anaerobic pathway, the glycolysis cycle. So there's a series of chemical reactions that happen in the mitochondria. Requires energy for this to happen. We break down glucose into a lot of ATP. We also have side products being built, and that's carbon dioxide and water in the process of these chemical reactions. So how do we store ATP? Gets stored in muscle, and then where do we get this glucose from? We can actually store it away in muscle fibers in the form of glycogen. So glycogen is a storage molecule, okay, which is just, again, these branches of glucose molecules bonded together, in fact, just like I'm trying to draw here. So excuse me for my crude drawing, but this would be glycogen. And what we actually do is when we want to break down a glucose molecule, we'll use an enzyme to break this apart, free this one, and then that one can be used to make that 32 ATP in this process. Okay. So when we talk about light to moderate exercise, this is exercise that would last, under 30 minutes, right? And again, not very strenuous. Okay, moderate exercise includes a light jog or a light walk, those types of things. But once we go to after that part, if it's, more than 30 minutes or if it's actually a strenuous activity, our body actually breaks down fatty acids. That's the main fuel after 30 minutes of exercise. And where do fatty acids come from? Lipids. And what is one of the lipids that we have? Glycoserides, like the fats that we carry in our body, in fact, can be used as fuel. And again, the key is after 30 minutes of that exercise, or if the exercise is very strenuous, we actually use fats to break down. All right, and that's actually, when you lose weight, you actually can lose fat. That's your goal, is to try to lose the fat, actually make your muscles larger, and that increases the tone, and this is being healthy, right? Okay, so this again just shows you, here's your glucose, converts to pyruvic acid, actually uses glycolysis to do that, and then we take that pyruvic acid from glycolysis to the mitochondria, and then the mitochondria will break it down and then make ATP. Notice that it can actually use lipids if it wants to break down a fatty acids, and we can actually use protein to do that. too. I want to mention that also, right? That proteins can be used for that. And this is not what you want to do, by the way. Use protein. You can, and that's a good survival method, but you really want to use this one over time, because that actually is actually what you want to do, right? It's most preferential. Protein, if you use protein, that actually means you use protein from your own body. For example, muscle is one of those. Okay, so again, during, for example, sports activities, right? Aerobic endurance, right? That means muscle contracting. We're using the aerobic pathway. Light to moderate activity, continue for hours, can use that method. And again, this is what you see, right? Just to kind of summarize what we're seeing, right? Short duration, ATP stored in muscle, gets used first, broken down, energy gets used from that. That next 10 seconds, ATP is made from creatine phosphate, can let us last longer there. 30 to 40 seconds or through the whole span of the end of the exercise, glycogen gets broken up. We use the glucose from the glycogen that generates the ATP. And again, this is anaerobic if it's a short duration, but if it's several hours of running, ATP gets used by many different energy nutrients, right? We can break down fat this way. All right. So what is muscle fatigue? Muscle fatigue is the inability to contract despite your stimulation. So what can cause fatigue? We need ions, of course, for this contraction to happen. Do you remember? Yes. Potassium gets used, sodium gets used, and calcium gets used in this whole contraction in different stages of the contraction, but the contraction nonetheless. You don't have enough ions, then you're not able to contract, right? So this should make a lot more sense, right? I believe that's how you spell Gatorade, but it's actually two words here. We come from this, right? Gatorade, right? And what is Gatorade mainly? Sugar and ions is basically what Gatorade is made up of. Sugar in the form of simple sugar, which gets broken down into glucose, and your ions like the ones mentioned. Right? So that's why Gatorade is so popular. That's why Gatorade can be beneficial if you are undergoing these long, strenuous duration types of exercises. Not for a daily drink, in fact. So drinking Gatorade, as part of your meal is not the way Gatorade was designed to be used for. Although it does taste great, is nice to drink, but Gatorade is not used for that. It's used for long hours of duration. And in fact, this comes from a special university, all right? The University of Florida. And it was actually a member of the football team, one of the staff members of the football team, that actually invented Gatorade. And in fact, if you look at the University of Florida and look at their mascot, it's the gator, right? So he used this solution, if you will, this drink that he invented to aid their Gator football team members. And that's actually where Gatorade, the word Gatorade, comes from. Interesting story. So go ahead and look that up if you want. But that's an interesting story to look at, and look at the actual person that invented that, which was part of the staff on the University of Florida football team. Right. So again, if you take away those ions, which is what he saw in South Florida, right? Or sorry, not South Florida, but the state of Florida, as because Florida's humid. In fact, you do a lot of sweat. So you're sweating a lot. You do a lot of ion loss, through sweat. Because of that, he was seeing many of the players fatiguing out, right? Their muscles were fatiguing. And so that's where this invention comes from. Drink Gatorade, and then you replenish your ions. You give yourself enough sugar to create enough energy for your muscles to continue to work, right? So this is where that comes from. Really interesting way to kind of tie all this together. Okay. So what else can happen, right? When we decrease ATP, we actually increase the magnesium levels. And actually magnesium can interfere with the T-tubule. And you know how the T-tubule, for example, the side of the T-tubule has those proteins that when they open, they release calcium. So that can happen as well. And glycogen, right? So our warehouse that stores all your glucose in the form of glycogen can be depleted, right? Okay, so for muscles to get back to where they're before exercise, right, the pre-exercise state, to get them back to normal levels, right, because most, exercise uses their, muscle contraction gets, used completely, and the muscles get very tired. And so how do we get that back, right? Well, we have to get oxygen reserves back in place, so we get those replenished. Lactic acid gets reconverted to pyruvic acid. Glycogen storage warehouses get replenished as well. ATP and creatine phosphate are resynthesized, and everything goes back to normal. And all these replenishing steps require extra oxygen, right? So we call this the excess post-exercise oxygen consumption stage. We use that word oxygen, or this statement oxygen depth, to talk about this, right? And this is the breathing, the heavy breathing that happens after a strenuous workout, right? So if you marathoners or joggers, once we get back to that level where we're, breathing a whole bunch, or when you run track or whatever you're doing, this breathing, if you will, is trying to get more oxygen in there to replenish everything to get it back to normal, all right?